All right, please confirm online people, am I audible? Everyone please confirm, am I audible? Okay, no problem. Let's continue. Okay. Others? Confirm everyone online, am I audible to all of you? Okay, right? Perfect. All right, let's continue. So now as all of you know that we are hardly having 8 weeks of time. Okay, so in these 8 weeks we have to complete 11 units, very effectively, which means that 1 week we have to complete, at least in 1 week we have to complete 1 to 1.5 units. Okay. So it is important, after 3 days onwards I'll take the classes up to evening. Okay, so we'll take the classes continuously. Okay, am I audible to everyone? Okay, all right, let's continue. So I'll take the classes continuously up to 4 o'clock. Okay, so from tomorrow please keep these timings. 9:30 to 10:30 will be paper 1, and 10:45 to 1 o'clock I will take the core paper. Okay, whereas from next week onwards, I mean to say from next Monday, 1 to 2 lunch break, and 2 to 4 again I will take the class, and 4:30 to 5:30 will have study hour. Okay, and because 1 week 1 chapter we have to complete. Okay, and every Saturday from morning to evening we will do the PYQ analysis of all the set as well as the net. Okay, and every Sunday you will be having exam. So remember, every Sunday 1 unit you have to write the exam, and every Saturday I will do the complete PYQ analysis. As a result, in the span of 8 weeks we can complete the whole syllabus. Okay, I want every one of you to come to the half line from tomorrow, otherwise I am telling seriously, I don't allow anyone to the classes. Okay, for whom sake we are taking the classes. Okay. Okay, all right. Okay. Now let's understand. So we'll start with the 2 units simultaneously. Now everyone remember, okay, this is the way where we continue the units. So we'll be starting unit 6, unit 1 biochemistry. We will do both simultaneously. Unit 6 and followed by the unit 1 biochemistry. Then we will go with the unit 8 genetics. Then we will go with the unit 13 techniques. Then we will go with the unit 12 applied biology. Okay, then we will come back to the most important ones, unit 2, then followed by the unit 7, right, followed by unit 11, unit 10, unit 3 and unit 4. Okay, so this is the order we will follow. Okay, so consider hardly 1 week we have to complete 1 unit very effectively. Okay, there is no problem. Whomever listening first time, try to understand. I will teach little slowly but effectively. Okay, see, this time whatever the points that I have missed in the previous sections, we will cover every single point. Only follow this PPT, do not follow the old PPT. Okay, and we'll solve every single PYQ. By that we'll get enough confidence of the topic. Right, so please continue everyone. We will be starting with the unit 6 as well as the unit 1 simultaneously. Okay, so let's start the unit 6, one of the most important unit in terms of the weightage. Okay, so what are the various subtopics that we have to focus in the chapter of unit 6? See, if you consider any kind of a set exam, any state set exam, we get 5 to 6 questions. Similarly, if you consider the net exam, definitely we get 5 to 6 questions. Okay, so the major concepts to be covered here is photosynthesis. In the photosynthesis, majorly we will focus on what? We will focus on the light reactions as well as the dark reactions. Then respiration is a different topic and photorespiration is a different topic. Photorespiration is nothing but what cycle? C2 cycle. Whereas in the respiration we will read glycolysis, Krebs cycle and electron transport chain, that we will include in the biochemistry, and will read the differences between plant respiration and animal respiration. And the third very important topic is what? Nitrogen metabolism. Why the volume is low? Volume is fine only. Okay. Am I audible? Yeah, okay. Perfect, perfect. Okay, now and in the nitrogen metabolism, most of the times the questions comes from the either genes or from the enzymes or from the bacteria or from the processes. In the entire plant physiology, one of the most toughest subtopic is plant hormones. It is tough because vast amount of the information to be remembered. I mean to say, whenever we are reading the plant hormones, we have to understand name of the plant hormone, from which source they have originated, what is the precursor involved in the biosynthesis of the hormone, their biosynthetic pathway, most importantly functions, applications, as well as the signaling process. The question comes from any corner. That's the reason we have to read the whole plant hormones without leaving anything. There is no way of getting the question paper without the question from photosynthesis and without the questions from plant hormone. So 200% we get the questions from here. Then followed by, there are most experimental based questions comes from the sensory photobiology, where most of the questions comes from cryptochromes, phytochromes, phototropins, long day plants, short day plants. These kind of questions will appear in the exam. And again, there is one more subtopic where questions comes very frequently in the exam is solute transport, especially the transport in the xylem and phloem. And again, there is a very specific unit. If you consider last 15 question papers, out of 15 times the question was appeared 15 times from the concept of secondary metabolites. Hence we have to read this concept from very basic to the very advanced. And interestingly, this is one of the most easiest topic. And then when it comes to the stress physiology, it is completely theoretical. We have to read and remember lot of information. Now, in the entire chapter of plant physiology, which are tough topics and which are scoring and easy topics, the tough topics are photosynthesis, then plant hormones. These two only the little tough. Of course, the respiration also. Whereas these three are only scoring, along with that transport in plants as well as the secondary metabolites. So how many subunits are there? We have 7 subunits. Out of 7 subunits, it is very important we all has to read 6 subunits compulsory. Why everyone is asking to increase the volume? Okay, just give me a moment. Volume is fine. Okay, all right. Okay, now let's start with the number 1 topic, which is photosynthesis. Okay, so there is no doubt, definitely we get one question from the photosynthesis. And the sad part is that how many number of times people may read the concept of photosynthesis, definitely they will give the wrong answer in the question. So whenever you are reading the photosynthesis concept, always remember my words, you have to read the concept with respect to the application based. Okay, if you don't read towards the application based, if you just read just like that, you never ever answer a single question. So always try to understand a deeper concept, especially application based. Okay, all we have to remember. Now let's start photosynthesis. Now tell me, how can we give a simple definition to the photosynthesis? What is photosynthesis? Preparation of food. So synthesis, to make something, to prepare something. What generally plant prepare? Plant generally prepare their own food, but they are preparing the own food by using light. That's the reason we called photosynthesis. In more simple words, photosynthesis is a process of production of food, or production of food, which is the sugars, by using sunlight as well as water and carbon dioxide. So carbon dioxide, sunlight, water, all are acting as what? Raw materials. So plants uses light, water, carbon dioxide, and with the help of chlorophyll. What plant is doing? Plant is reducing. The question in the set exam. See, do not confuse, you will understand in the upcoming slide. Production of sugar is always a reduction reaction. But overall photosynthesis is oxidation and reduction. But production of sugar is always reduction, because we are reducing the carbon into glucose. Correct na? Hence it is a reduction reaction. Whenever plant is preparing its food, what the plant is doing? Plant is releasing oxygen as their byproduct or waste product or end product. None of the plant never ever release the oxygen purposely. Whenever plant is preparing its own food as a byproduct or end product, they are going to release the oxygen. That is the oxygen what we generally breathe. So we read the equation from the 9th standard, the most simple equation. What is that? 6 molecules of, say simply remember 6, 6 and 1, 6. Correct na? Which means, we'll do it in the next class, biochemistry. 6 molecule of carbon dioxide combined with 6 molecules of water, we are getting 1 molecule of sugar. So, but what is the point? Carbon dioxide is available, water is available, sunlight is available. But even though these 3 components are available, the most important component is what here? Chlorophyll. See, if carbon dioxide is available, water is available, light is available, if the chlorophyll is not present, again photosynthesis never takes place. Consider there is no carbon dioxide, but abundant amount of water is present, light is present, and chlorophyll is also present. That time also glucose production never happens. If the glucose wants to be produced, definitely we require carbon dioxide, water from the soil, carbon dioxide from atmosphere, light from the sunlight, and chlorophyll. When all are present, then only plant can reduce the carbon into sugar. Then there is a release of oxygen. It's a very simple thing. Now, in the competitive exam, at least one option or one question directly comes from where. Where represents what? Where that specific reaction is taking place. Means knowing the location of the reaction is utmost important towards the exam point of view. In a given 4 options, at least one option comes from the location. Now, where does the photosynthesis takes place? Chloroplast. What is chloroplast? Chloroplast is a simple double membrane cell organelle. Correct na? Where this chloroplast is present? Is it present in the plants or not? So all the green plants generally contains what? Chloroplast. If you observe the structure of a chloroplast, what does it contain? Chloroplast is a what? Double membrane organelle. Correct na? It's a double membrane organelle which is called as outer membrane, inner membrane. The space between the outer and inner membrane we called intermembrane space. Now, if you observe inside the chloroplast, what we can see here when we observe inside the chloroplast there are flattened vesicle-like structure. You can see here. So exactly like this. This flattened vesicle-like structure, what do we call it as? Thylakoid. So this is one single thylakoid. But 90% of the time, thylakoids are never ever arranged as a single. Instead of arranging in the single manner, they are always arranged on the group. So in simple words, a group of thylakoids. We generally call it as what? Granum. Now again, one group of thylakoids can also be connected to the another group of the thylakoids. How? What you can observe here? This is one group of thylakoid and this is another group of the thylakoid. One group of thylakoid can be connected to the another group of the thylakoid. What you can observe? By a thin thread-like structure. Correct na? This thin thread-like structure which is connecting two granums, we generally call it as what? Lamella. Okay, it's not lamellae, it's a lamella. So ends with A. Now, what else you can see in the chloroplast? We have a thylakoids arranged in the form of a granum. Now, where all these thylakoids and where this granum is present, see, in a simple words, when we observe inside the cell, most of the space is occupied by cytoplasm. How does the cytoplasm look like? It is not a solid. It is not a liquid. What it is a gel. It's like a semi-solid, like ointment texture, right? Similarly, inside the chloroplast, we have a, what is that? Gel-like structure, correct na? Or matrix-like structure, which we call it as stroma, right? What else it contains? You can observe the chloroplast and mitochondria are what kind of organelles? Semi-autonomous or self-replicating. What does it mean? They are having own DNA and own RNA and own enzyme. Hence, they can synthesize their own proteins. What about the other organelles? Other organelles generally import the protein from cytoplasm. Correct na? Whereas, chloroplast and mitochondria also import the proteins from cytoplasm definitely, but some of the proteins they can synthesize on their own. That's the reason we call self-replicating or semi-autonomous organelle. So definitely they are having ribosomes. What kind of ribosomes they contain? 70S. Now we all know very well there are two type of ribosomes, 70S and 80S. Where 80S are present in? Prokaryotes or eukaryotes? Eukaryotes. And the chloroplast is also in eukaryotic, it is present in eukaryotic cell. But why chloroplast contains 70S ribosome instead of 80S? It is derived from? Very good. So according to endosymbiotic theory, it was understood that mitochondria and chloroplasts are derived from the external membrane of the bacteria. That's why even though it is a eukaryotic cell, the type of ribosomes are always 70S only. What else it contains? We can observe it contains large starch granules. Starch granules are not smaller. They are large in size, less in number. But you can observe something called yellowish one. We call it as what? Lipid droplets. They are small in size and more in number. Right. So let's quickly understand what does it contain. Chloroplast is a double membrane organelle which contain outer membrane and which contain inner membrane. Then what else it contain? It contain a flattened vesicle-like structure called as thylakoid. One group of thylakoids are generally called it as what? Granum. One group of thylakoids can be connected to the another group of the thylakoids by a thin thread-like structure called as lamella. The fluid or gel or a matrix-like structure called as stroma. And the type of ribosomes are always 70S and it contains lipid droplets which are small in size which stores lipid. And what this starch granules will store? Abundant amount of the starch. Now the most interesting one, this chloroplast having different shapes in different organisms. What's the most common shape? So the most common shape is oval, and in some cases it look like a biconcave. In some organisms the chloroplast look like a dumbbell shaped structure, right? In different organisms it is having different shapes. And the most important statement we all people has to remember, the chloroplast contain a specialized DNA which is called circular double-stranded DNA. Correct now, it is not a single-stranded. The most important statement here, the double-stranded DNA present in the chloroplast, it is coding for an important protein. What is the name of that protein? Rubisco. What is Rubisco? Rubisco is the most abundant protein on this planet Earth. Why we use the word abundant? See, Rubisco is an important protein synthesized by the DNA of the chloroplast. But why it is abundant? Because if you take any leaf of any plant on this earth, 40% of this leaf contains Rubisco. Since Rubisco is present in every leaf of every plant present on the earth, it is present more abundantly. That's the reason we called abundant protein. In our body, which is the most abundant protein? Collagen, because it is also present everywhere. Example, tendons, ligaments, joints, arteries, uterus, everywhere it is present. Since it is present most abundantly, we call it as abundant protein. right? This is the simple structure of the chloroplast. Now it is important to understand inner view or the deeper structure of the chloroplast. Why we have to read inner view or microscopic structure of the chloroplast? As I said, different reactions happens at different places. That's the reason we have to understand outer structure as well as we have to understand inner structure also. We understand flattened vesicle like structures are called as thylakoid. So this is how a thylakoid look like. And what we understand, all the thylakoids are always arranged in the form of a bundle or a stack, which is called as granum. Correct na? If you observe one thylakoid separately, every thylakoid having what? Every thylakoid having a membrane. Correct na? Nothing but plasma membrane. So the plasma membrane of this thylakoid we call it as lamellae. It is not lamella. What is lamella? One group of thylakoids connected to the another group of thylakoids by a thin thread-like structure. This is called lamella. What is lamellae? The outer membrane of the thylakoid called as lamellae, right? And in simple words, you call thylakoid membrane, also called as lamellae. Now, any organ having an internal space. You remember, we read something called as blastula. Blastula contain inner space. What do we call it as? Blastocele. Correct? A fluid-like structure. Similarly, the thylakoid also having an internal space. That internal space of this thylakoid we called lumen. Why we have to read about the lumen? Why we have to read about the membrane? Because different reactions are happening at different places, which means in the lumen different reaction occur. An example, we will see that breakdown of water. Correct na? What reactions happen on the membrane? Light reactions are strictly happening on the membrane. If you take stroma, dark reaction. So in a different parts of the chloroplast, different reactions will take place. That's the reason we have to read the outer view as well as the inner view also, right? Now understand. So everyone focus. I want every one of you to solve every single PYQ which we are going to discuss on this Saturday, okay? Hence I want you to listen, okay? Do not think that you already know the concept, okay? Understand the points very correctly. What is the ultimate goal of the photosynthesis? Remember, simple words. Ultimate goal of a plant is reduce the carbon into sugar. This is ultimate goal. See, why plant is struggling ultimately to prepare their food. How they can prepare the food? Just by reducing the carbon into glucose. Now the question is here, plant can't reduce the carbon into glucose very easily. Why? It is extremely a complicated process. Plant cannot take the carbon dioxide from atmosphere and just directly it cannot reduce into the glucose. Since it is a very typical process, plant has to produce energy first. If they produce energy, fixing the carbon into glucose is easy. Correct na? So what is the meaning of production of energy here? Production of ATP and production of NADPH. What is ATP? Universal energy currency, energy equivalent. And what is NADPH? NADPH is what? Nicotinamide, adenine, dinucleotide, phosphate. Correct na? Now my question is here, NADPH is also giving rise to what? Energy only. Then why I cannot call NADPH as energy equivalent? Lakshman, why we can't call NADPH as the energy equivalent? The reason is very simple. NADPH cannot give rise to the ATP directly. It is an indirect form of the energy. Hence you cannot call it as energy equivalent. ATP means you are getting the energy directly. Correct na? Moreover, NADPH not only giving the energy, but it is also acting as what? Reducing equivalent. The most important term I want all of you to remember whenever you come across NADPH or FADH, to remember a common point. They are generally act like electron carriers. Correct na? They are involved in the transport of electron. Whenever electrons are transported, then only energy can be generated. Correct now? That's why they are called reducing. Why we call reducing equivalent? Upon gaining the electron, they become reduced. That's why we call reducing equivalent. Then why we cannot call ATP as the reducing equivalent? Because it is never involved in the transport of electron. Its function is only what? Production of energy. That's why ATP is energy equivalent, NADPH is reducing equivalent. Correct na? So these two generally produced during the light reaction. What does it indicate generally? If I want to produce the light, definitely what I require, if I want to produce the energy, definitely what I require? What I require? Light. Come. So without light, it is impossible to produce energy. Correct na? So now don't confuse with my statement, all of you continue. We are doing photosynthesis, continue. Now look into this, all of you. What I want to do here, what is the ultimate goal of photosynthesis? Reducing the carbon into glucose. In order to reduce the carbon into glucose, I don't require light. You're getting my point? I don't require light. When I require light, I require the light to produce energy. What's the function of energy here? If energy is there, then only I can reduce the carbon into glucose. You're getting the point? That's the reason photosynthesis is broadly categorized into light reactions and dark reaction. So ultimate goal of the light reaction is to produce energy equivalents. Correct na? Ultimate goal of dark reactions is what? To reduce the carbon into glucose. That's all. Now when does the light reactions takes place? During the day or night? Day. What is the reason? Because ultimately we require the light to produce energy, because ultimate source of energy in this universe is light, sunlight. So if I want to produce the energy, definitely I require light. Hence light reactions only takes place in the presence of light. And what about the dark reactions? Day or night? Night. See, remember, dark reactions doesn't mean that it is occurring in the dark. Dark reactions means that light independent. Correct na? See, what is independent here? Whether the light is present or absent doesn't matter. What should present? Energy. Correct na? But in order to produce the energy, we definitely require the light. That's why dark react, because simple. Plant can produce the food morning time also, plant can produce the food evening time also. Correct na? Hence reduction of carbon can happen in the day also and night also. Right? Then why did we use the word dark? We used the word dark, there is no requirement of light. That's the reason we used the word dark reaction, right? So ultimately we have to understand very clearly how does this light reactions happens, then it is easy to understand dark reaction. In the previous 2025 June, whatever the exam you have written, they asked a question from dark reaction. In the 2024 paper, they asked a question from light. In the before exam, they asked questions from both. Okay, of course it is so important, we have to understand everything very clearly. Okay, photosynthesis is 90% conceptual, 10% you have to remember. Now let's focus on light reaction. Do not forget till the end, our ultimate goal is what? To produce ATP and to produce NADPH. Okay, so now first point, we already discussed in our exam, most of the questions will comes from where? Right. So where does this light reactions are taking place? Where they are taking place? Thylakoid membrane or lamellae. Now not lamella, lamellae. Okay. Now the question is why? Why light reactions are only happening on the thylakoid membrane? Why not in any other areas? See, what is the ultimate goal of light reactions? To produce energy. In order to produce energy, we require photosystems. We require a lot of electron complexes, where all these electron complexes and photosystems are present on the thylakoid membrane. Membrane nothing but made up of lipids only, right? So what you can observe on the membrane, what we are having? We are having photosystem. Photosystem 2, photosystem 1, and variety of electron acceptor complexes. We'll see that. Plastoquinone, cytochrome B6F, plastocyanin, and the most important enzyme. What is that? F0 F1 complex. We call it as ATP synthase enzyme. Where it is located, all are located on the membrane itself. That's why light reaction strictly happens on the membrane. Now see the interesting point, we'll see that, we'll understand in the upcoming slide. This is what region? Stroma. This is membrane. And what about this? Lumen. Wonderful. This is what? Lumen. Now look at this slide and try to understand. By using variety of photosystems and electron acceptors, we are producing energy. Energy such as ATP and, but can you observe something? ATP and NADPH, they are releasing which side? Stroma side. Correct now? Why it is releasing from the to the stromal side? Because dark reactions happening in the stroma. So the production starts at the membrane, but releasing happens onto the stroma. Correct now? And observe the picture. We'll see that in the process of the light reactions, there is a breakdown of water through which oxygen will be generated. We will see that. But where this breakdown of water is happening? Lumen. So it is very important to understand variety of reactions happen at different places. That's the reason it is important to understand the locations. Now let's focus light reactions. Again, understand our ultimate goal of the light reaction is to produce energy. So ultimate source of energy is what? Sunlight. See, sunlight comes continuously. So someone has to absorb that light energy and has to convert it into chemical energy. Then only plants or any other organism can utilize. Now, who is going to absorb the energy from the sunlight? Who will absorb? Who will absorb? Pigments. Correct na? So in the plant, there are many pigments are there. Okay, so there is a great possibility from last 2 to 3 times the question was not appeared from the pigments. There is a possibility this time we can expect. Okay, all the pigments are broadly categorized into principal and accessory. Sometimes the accessory pigments are also called as antenna pigments. Right. Now tell me why we call principal the most important. That's why we call principal pigments. Okay, principal pigments are very important pigments, but they are what we can say, they are abundant in number. Abundant in number. Okay, best example for the principal pigments, chlorophyll A. We have variety of chlorophyll, chlorophyll A, B, C, D, E. But when we consider all the higher green plants, in the higher green plants we have majorly chlorophyll A. That's why in almost all the plants, chlorophyll A said to be what pigment? Principal pigment. Correct na? Where it is mostly present in the green plant. So bacteria perform photosynthesis or not? Bacteria. Of course, yes. Bacteria also perform the photosynthesis because they are also having pigments. So they can also absorb the light and perform the photosynthesis. Algae performs or not? Algae also perform. In more simple words, the organisms which are having the chlorophyll pigments, they can perform the photosynthesis. Now, the same chlorophyll A, it is also present in purple bacteria. But we can't use the word chlorophyll A. It is, we'll see that, it is structurally modified. So how can I give the definition here? The structurally modified chlorophyll A present in the purple bacteria called as bacteriochlorophyll A. Similarly, the modified chlorophyll A present in the green sulfur bacteria, which modified chlorophyll A present in the green sulfur bacteria called as chlorobium chlorophyll. Generally what? All the organisms contain chlorophyll A, but it exists in a different forms. But only in the higher plants it exists in the normal form or in the original form. That is the reason all higher plants contain chlorophyll A as the major pigment. Chlorophyll A shows bright or blue-green color. Blue-green, you can't use the word green, okay? We can use the word blue-green. But it reflects which color? Reflects. See, chlorophyll A reflects green color. Why plant appears green in color? Because they are reflecting green color. Whichever the color absorbed by the pigment, it never appears in that color. Whichever the color it is reflecting, That's why it appears in green in color. You take a carrot. Carrot looks in orange color because it contains pigments that reflects orange color. Correct na? Here it shows brighter blue-green color because it is reflecting that green color. That's why it appears in the green color. I hope everyone is getting correctly. Now, these are all pigments are said to be what pigments? Principal. Most important for the plant function. Then we have accessory and antenna pigments. No need to confuse. They are also extremely important. There is no doubt about it. But compared to the principal, they are less in number. And very interestingly, all the accessory pigments or antenna pigments are of different types. See here, chlorophyll B, C, D, E. They are all not present in the higher plants. All the higher green plants only contain chlorophyll A, whereas chlorophyll B, C, D, E is present in the lower plants such as green algae. Since it is not present in the green plants and it is not a major pigment, we call it accessory or supporting pigment. Don't think that they won't absorb the light. They absorb the light, but compared to them, plant prefers chlorophyll A. Green algae, instead of the chlorophyll A, they prefers these pigments. Right? They are having their own importance. And next accessory pigment is what? Carotenoids. No need to get confused here. Carotenoids are of two types. One is called carotenes, right? Another one is called xanthophylls. There are two types of carotenoids. What is called carotenes? The carotenes are just made up of only two atoms, carbon, hydrogen. That's all, nothing else it contains. I will show you the structure next slide. What about the xanthophyll? Along with the carbon, hydrogen, it also contains oxygen. That's all. That's a simple structural difference between these two. So, what are the example of the carotenes we read? Beta carotene. Where it is present? In the carrot. What is the name of the carotenoid that is present in the tomato? Tomato contain a pigment named as lycopene, which reflect red color. That's why tomato appears in red in color. Correct na? And so these two are the examples of carotenes. What are those? Beta carotene and lycopene. Examples of carotenes. Then we have xanthophyll. So xanthophyll, the standard example, please remember, lutein. It is also a type of carotenoid oil. Okay. Now the third one, phycobilin. So when you observe the phycobilin, remember phycobilins are majorly found in cyanobacteria and rhodophyte. Only in these categories we can get most of the phycobilins. There are a common example of phycobilins are phycoerythrin, phycocyanin, yellow phycocyanin. There is nothing to explain. I want you to remember the examples definitely. Okay. Now, the common point all of you has to remember, whether it is a principal pigment or an accessory pigment, the common function is what? Absorbing the light. Now the question is here, if all the pigments are involved in absorbing the light, what is the use of all the pigments? Any one is sufficient, because this is also absorbing the light, these are also absorbing the light, everything is absorbing the light. Then why plant require all? Why plant require all? Correct. Because they absorb the light at different wavelengths. Correct na? Chlorophyll A absorb the light at a different wavelength and B will absorb the light at a different wavelengths. Correct na? Apart from this, there is one special pigment. What is that? Carotenoid, which protect the plant from high intensity of sunlight. An example, when we expose to the high intensity of sunlight, we will escape because we can move. We can flee, means we can move. But plants cannot escape. Plant definitely has to tolerate to the high intensity of sunlight. How they can tolerate? With the help of carotenoids. So carotenoids are said to be photoprotective pigments, which protect the plant from higher intensity of sunlight. Chlorophyll won't do that. Chlorophyll function is what? Only for the absorption of the light. Correct na? Whereas phycobilins are also absorb the light majorly. They also won't act like a photoprotective pigments. Now What are the pigments we read now? We read chlorophylls A, B, C, D, E, carotenoids and phycobilin. The most common question they used to ask in the exam, all of you answer, among all the pigments, which pigments are water soluble? All of you online people answer, among all these pigments, which pigments are water soluble? Niranjana, what are water soluble? How come you forgot, huh? This many times I thought. Everyone. Neelima, which are water soluble pigments? Okay, I'll explain the structure. Now you people tell me. Okay, everyone online people. All right, look into this. Now it is super important. We have to remember the structures mandatorily for the exam. Okay. There is a great possibility questions can comes from the structural features. You no need to remember the structure. You have to remember structural feature, what exactly it made up of. Now let's focus chlorophyll. Can you observe this is the structure of the chlorophyll? Chlorophyll, very good. Chlorophyll contain two portions. One is called head, another one is called tail. The head of the chlorophyll we call porphyrin head. What head? Porphyrin head. The tail of the chlorophyll we called phytol chain. Okay, head of the chlorophyll called porphyrin, tail is called as what? Phytol chain. Now question is very clear, why did I call it as porphyrin? Why did I call it as phytol? Observe the pictures very correctly. How can I define the word porphyrin? Look into this. It contains variety of pyrrole rings A, B, C, D, E. Can you see that? They are all said to be pyrrole rings. How many pyrroles? There are 5 pyrroles are there, but 4 pyrroles, that is A, B, C, D. All 4 pyrroles are coming close together. Correct now? How they are coming close together? Can you see? Via what linkage? Nitrogen. Can you see that? All the 4 pyrroles coming close together towards the center with the help of nitrogen and surrounding the central metal atom magnesium. That structure called as porphyrin. So porphyrin is simple words, it's a tetrapyrrole. Correct now? How many pyrroles are there? 5 are there, but 4 are surrounding the magnesium. A tetrapyrrole ring come close together and surround the central metal atom magnesium. You remember, we were continuously saying a word that chlorophyll absorbs the light. Correct now? How it will absorb? Chlorophyll having what center? Magnesium. Remember all of you. Magnesium is the one act like a chromophore. Chromo color. 4 which absorbs. So since with the help of magnesium only porphyrin head absorbs the light. Okay. What about the other part? Phytol. So simple it is. Can you observe? Phytol is a very long hydrocarbon chain. Nothing else it contains. A long hydrocarbon chain. Correct now? Now the question, no need to get confused, understand. So it is super clear that head contain magnesium. That's the reason it can absorb light. Now what's the function of the tail? Now observe, in the upcoming slide you will get a brilliant idea. This is what membrane? Thylakoid membrane contain what? Lipids. Correct na? So the tail will embed the head onto the membrane. You're getting? Consider this is only the chlorophyll. This is what? Head. This is tail. Now it exactly incorporate like this. So then head is exposed outside. Correct na? Then head starts absorbing light. So what's the function of tail? Tail help the head to embed into the membrane. Right? That's the simple structure of the chlorophyll, containing porphyrin head and phytol chain. Very good. Next, now just now we discussed we have a chlorophyll A, B, C, D, E. And what is the common point? The common point is that every single chlorophyll having porphyrin head and phytol chain. Now how can we differentiate one chlorophyll molecule with the another chlorophyll molecule? Exclusive, of course, based on their wavelength, but here we are talking with respect to structure. How can we differentiate one chlorophyll with the other chlorophyll? Exclusively based on their side chain. Correct na? If you observe the chlorophyll A, it is having methyl group as their side chain. Correct na? If you observe the chlorophyll B, it is having aldehyde. Bacterial chlorophyll having something else. So one chlorophyll molecule can be differ from another chlorophyll molecule exclusively with the help of side chain in terms of structure I am talking about. So now carotenoids. What you people can observe about the carotenoids? So can you observe carotenoid? It's a 5 carbon ring with alternate single bond, double bond, single bond, double bond. A 5 carbon containing compound with alternate single bond and double bond. This is called carotenoid. This structure generally called as isoprene. Okay, isoprene is what? A 5 carbon containing compound with alternate single bond and double bond. Then only we call it as what? Carotenoid. So simple. Then what is the next one? Bilin pigments. Just now we all discussed, right? See what you people can observe in the bilin pigments. They are also having what chains? 1, 2, 3, 4, which are pyrrole. What is the difference between these pyrroles and these pyrroles? Here all the tetrapyrroles are coming towards the center. Here they are straight chain. Hence this is called linear tetrapyrrole. Correct na? So a linear tetrapyrrole is called as bilin pigment. Carotenoids are isoprene. What about this? Chlorophylls are porphyrin head and phytol chain. Now all of you please answer. Now what is the question I asked? Among all the pigments which are water soluble, why not chlorophyll and carotenoids? Wonderful, very very good. They contain what? Hydrocarbon chain. Hydrocarbons are what? Lipid in nature. Correct na? Hydrocarbons. Correct na? They are lipid in nature. Right? That's the reason chlorophyll and carotenoids are strictly lipid soluble. The only pigment which is water soluble is what? Bilin pigments. Okay, clear. Now again the most important slide, n number of times the question were asked exclusively from this particular slide. Just now we discussed that all the pigments are going to absorb the light. Correct na? So based on the absorption of the light, there were 2 spectra were derived. One is called absorption spectra and action spectra. I will tell you in a simple words, let's differentiate it very correctly. Absorption spectra, it is always talking about pigment. Okay. Action spectra always talks about process. What they talk about? Process. Now let's understand it elaboratively. So in very simple words, what exactly absorption spectra explain? At which particular wavelength a particular pigment absorbs the light. Consider I am a chlorophyll A. At what wavelength I am absorbing the light? That is my absorption spectra. Now let's understand it very correctly. Okay. Focus on the picture correctly. Absorption spectra. On the x-axis what we have taken? Wavelength. On the y-axis we have taken amount of the light absorbed. Now all of you please focus on the picture. I have taken only 3 chlorophylls, 3 molecules or 3 pigments. One I have taken chlorophyll A and chlorophyll B. The yellow color one is called as what? Carotenoids. Now all the students focus and understand my words very correctly. Remember this statement. Whatever the color absorbed by the pigment, it never appears in that color. Whatever the color opposed or reflected by the pigment, it appears in that color. You are getting the point. Now let's focus on the picture very correctly. I don't know what is the problem with the volume. Every time it is fine only. Okay, anyway, now I can't change it. After the class, very surely I'll look into it. Okay, all of you focus on this picture correctly. See, first let's consider the chlorophyll A. Okay. I am considering the chlorophyll A first. Can you observe? This is the peak of the chlorophyll A. See, this is the peak of chlorophyll A. What you people can observe? The peak is very high at dark blue light and light blue light. Correct na? the peak was drastically fallen down when it reaches to which light? Green light. Correct na? So what you can observe now, the chlorophyll A can easily absorb dark blue light and light blue light, but it is reflecting green color. So since it is absorbing dark blue light and light blue light, they never appear in blue color. Since the chlorophyll A is reflecting which color? Green color. That's why plant appears to be green in color. Correct na? Now look into the carotenoids. What you people can observe by the carotenoids in the graph? Carotenoid graph is this. What you can see here, it can absorb dark blue light, light blue light, it can also absorb green light. So it never appears in these three colors. Now you take a best example of carotenoids present in the carrot. Carrot, it contain what? Beta carotene. Correct na? So beta carotene is the type of the carotenoid. It is reflecting which light? Orange light. That's why carrot appears to be orange in color. The same type of carotenoids are also present in tomato, which reflects which color? Red color. That's why tomato appears to be in red color. So in very simple words, whichever the color absorbed by the pigment, it never appears in that color. Whichever the color reflected by the pigment, it appears in that color. What is absorption spectra? The point at which a particular pigment is absorbing the light. In simple words, the maximum absorption shown by the pigment, call it as absorption spectra, right? Now, what is called as action spectra? Action spectra never ever talk about pigment. It is always talking about a process. What does it mean? At which specific wavelength, what is the point? At which specific wavelength a particular process is happening. You take any process as an example only. You take an example of photosynthesis only. What is the maximum light that is required for the process of photosynthesis? 600 to 700 nanometer is the idealized wavelength for the process of photosynthesis to occur. So what is action spectra? The point at which a specific process is happening continuously. What is that? Action spectra. So 600 to 700 nanometer. Why? Because at the 600 to 700 nanometer, the pigment starts absorbing maximum light required for the process of photosynthesis. That is what exactly we call action spectra. In very simple words, action spectra explains what is the exact wavelength required for the specific process to occur. What is absorption spectra is explaining? What is the maximum wavelength absorbed by a pigment? That is called as absorption spectra. You have to remember that very correctly. Okay. Now, how many photosystems? Still we are reading what? Light reactions only, which is a very broader area. What is the ultimate goal of light reactions? Don't forget this. Production of ATP and production of NADP. Correct na? That is the ultimate goal. Okay. Still we have not produced any of this. What we observed, in order to produce the energy, someone has to absorb the energy. Who is absorbing? Pigments. So that's why we were talking about the pigments. Now we understand absorption spectra, action spectra, and different types of pigments. Now let's focus how exactly we are going to produce ATP and NADPH through the light reactions. How many photosystems are there in the plant? Higher green plants always having two photosystems. Correct na? PS1 as well as PS2. Photosystem 1 and 2. Why we have given the name PS1 and PS2? Why we have given the name PS1 and PS2? It was just given based on the order of discovery. This was first discovered, this was second discovered, that's all. What's the common point we discussed? All higher green plants generally contain which pigment? Chlorophyll A. Correct na? Now we read that photosystem 1 is called as P700. Photosystem 2 called as P680. Why? What is the reason? See, what does it contain? Every photosystem contain what? Chlorophyll A. In the photosystem 1, the chlorophyll A absorbs the maximum light at 700 nanometer. That's why it is called P700. Similarly, in the photosystem too, what's happening? The chlorophyll A only it also contain. It absorbs the maximum amount of the light at 6,8. That's why we call P68. Okay. And again, the question asked in the exam, don't get confused. Now, can I commonly use one word? PS1 and PS2 generally contain chlorophyll A. Yes or no? Yes. It is common. But the point is here, in the PS1, compared to the PS2, more chlorophyll A's are present. This is the line which was asked in the exam. High chlorophyll A to B ratio. Where? In the photosystem 1. But if you observe the photosystem 2, equal amounts of chlorophyll A is present as well as chlorophyll B is present. Right? That is one of the most important difference to be remembered. Okay. Now, photosystem 1 majorly present where? In the green sulfur bacteria. Right? Whereas photosystem 2 majorly found in the purple bacteria. It is super important. I want you people to remember the examples compulsory. Switch on the AC, man. Okay. Now, observe, see the next 3 slides whatever we are going to discuss is the most important slides to help you understand the light reaction. If you understand these 3 slides, whole concept of light reactions is super clear. Okay. So, observe the pictures very correctly without having a confusion. Okay. Observe all of you. Okay. These 3 are very important and crucial slides to understand. This is what membrane? Thylakoid membrane. Correct na? Now observe. Okay. Just imagine like this. This is a thylakoid membrane. On the thylakoid membrane we have photosystem. Correct na? What actually photosystem is made up of? Remember, photosystem generally made up of a protein complex named as LHC. What is that? Light harvesting complex. What is the meaning of harvest? Which can absorb and transfer the energy. So, every photosystem contains what? LHC. Every LHC made up of 2 components. Every LHC made up of principal pigment and accessory pigment. Wonderful. Correct na? Principal pigment most of the time we call it as reaction center. Antenna pigments we already studied, now also called as accessory pigments. You are getting the point? Once again all of you try to understand. So, consider this is only thylakoid membrane. Correct na? This is what now? Photosystem. Photosystem contains LHC. LHC made up of? So, can you see this? This is called principal pigment. Now, what are all these? Accessory pigments or antenna pigments. So, that is the simple composition of the photosystem. Now, let's prove very clearly how does this process of ATP is getting synthesized. All of you focus on the picture very correctly. This is the most important slide to understand for the exam. Now, can you see this dark green color one? What is this? Reaction center. Very good. Correct na? Reaction center is also called as principal pigment. Now, what are all these light green color ones? Antenna pigments are accessory. Made up of? We discussed na, principal pigment is only made up of chlorophyll A. Antenna pigments are made up of? Chlorophyll B, C, D, E, carotenoids and phycobil. All together called as antenna pigments. Only chlorophyll A is present in the reaction center. Now, what's happening? Observe. Observe the picture very carefully. Now, first, what is the function of every pigment absorbing the light? Correct now? Now, observe the concept very correctly. First, all the antenna pigments absorb the light. Correct now? Upon absorbing the light, they are transferring the energy from one pigment to the another pigment. Correct now? They are transferring the energy from one pigment to the another pigment, which is called as resonance transfer of energy. Passing the energy from one pigment to the another pigment. Finally, all the energy reached where? Reaction center. Consider I am a reaction center now. All of a sudden I have gained abundant amount of energy. Then what happened? I get excited. Upon excitation, I am going to release an excited electron. Correct na? Why I released this? Because I get abundant amount of energy, so I got excited and release one electron. Now this electron is accepted. So someone has to accept the electron. Who will accept? On the thylakoid membrane we have several electron acceptors. What? We have several electron acceptors are located where? On the thylakoid membrane. I believe everyone is getting. Now I am talking about photosystem, is it? Photosystem contain LHC. LHC contain antenna pigments. They absorb the light, send it to reaction center excited, release the electron. Now where it passed that electron? It passed the electron to variety of electron acceptors. So many electron acceptors are located on the membrane. Correct now? Now what is happening? Whenever electron is moving from one electron acceptor to the another electron acceptor, there is a loss of energy. You are getting the point? There is a loss of, see, now I release electron, I pass it to her, she is passing to hold the glass. Then what happened? Electron is moving continuously, hence it lost the energy. That energy can be used to produce ATP. What's the ultimate goal of the light reaction? To produce ATP and NADP. So how we are producing the ATP? By the resonance transfer of energy. Let's understand that little more elaboratively. If you understand these slides, then only concept is clear, otherwise it will be super confused. So antenna pigments absorbs photons and transfer it to the reaction center, which is called resonance transfer of energy. Then who is getting excited now? A reaction center get excited, releasing excited electron or high energy electron. Now that electron is passed through the series of electron acceptor. Whenever it is passing from one electron acceptor to the another, what's happening? There is a loss of energy. That energy can be used to produce ATP. Now we'll understand who are these electron acceptors, how this energy or ATP can be synthesized. Now focus, this is important, understand. So the process of formation of the ATP called as phosphorylation. In very simple words, phosphorylation means, friends, joining of ADP with PI. Correct na? Adenosine diphosphate with inorganic phosphate. Now tell me, can we add easily? No. What do we require now? Energy. I hope you are getting my point. If I want to join ADP and PI, then what I will get? You are getting the point. How can I join? In order to join, what I require? Energy. Now how the energy is coming from? When electrons are moving from one electron acceptor to the another, there is a loss of energy. That energy we used here to combine with ADP. Then there is a formation of ATP. So production of ATP called as, since every entire process or entire energy comes from light, that's why this process is called as photophosphorylation. Now focus from here correctly and solve the questions. Photophosphorylation is of two types, cyclic and non-cyclic. Why we use the word cyclic? See, electron started from here and electron reached back to the same position. Means the origin and destination, starting point and ending point is same. That's why cyclic photophosphorylation. Remember, it never ever happens in the plants. It only happens in the bacteria. Okay. In bacteria, there are no two photosystems. In bacteria, there is only one photosystem. Only one photosystem is involved here. Whereas, what is called non-cyclic photophosphorylation? The starting point and ending point, both are completely different. And non-cyclic photophosphorylation strictly happens in the higher plants. How many photosystems are there? Two photosystems are involved here. Now let's focus on it very correctly. This is the concept which we have to remember compulsory. So focus on cyclic. You should never ever compare cyclic and non-cyclic. Why you should not compare? This is happening in bacteria, one is happening in the plants. In order to understand, in order to remember only we compare. Correct na? While solving the question you should not compare. Okay. Now because both are completely different. Now let's focus cyclic. How many photosystems are there? Only one. Okay. This is only photosystem, whatever I am drawing, this is photosystem 1. Made up of LHC. LHC contain antenna pigments and principal pigment. Now observe the picture, all of you. What's happening now? Antenna pigments absorb the light and transfer the energy to the reaction center. Reaction center get excited now, release the highly energy electron. So someone has to accept it. Who is the first electron acceptor? FD, ferredoxin. The first electron acceptor in cyclic photophosphorylation. After FD, PQ. PQ represents plastoquinone. There are two types of quinones, plastoquinone A and plastoquinone B. After that, electron is coming to cytochrome B6F. Correct na? From cytochrome B6F, electron is coming to where? Plastocyanin. And from the plastocyanin, electron coming back to the reaction center. That's the reason the starting point and ending point is absolutely similar. That's the reason we call it as what phosphorylation? Cyclic photophosphorylation. There is no confusion over there. What is important for the exam point of view? You have to remember electron acceptor. Number 1, FD, plastoquinone, cytochrome B6F and plastocyanin. Correct now? Now the next type of photophosphorylation is non-cyclic. You should never compare. That is completely over. Happened in the bacteria. Now this is non-cyclic. How many photosystems are there? 2. P680 and P700. Now when you observe this picture, so what is non-cyclic? Starting point, ending point is different. Correct na? 2 photosystems are involved. Can you see here? This is the movement of the electron transfer. When we observe the non-cyclic photophosphorylation, it seems to be a sleeping jet. Correct na? The electron flow look like sleeping jet. That's why it is called jet scheme, or also called as zigzag scheme. There is no confusion. All of you focus on the picture correctly. Now what? So observe again, who is absorbing the light first? This is P680. Correct na? P680 contain what pigments now? Antenna pigment. And what is this? Reaction center. Very good. Antenna pigments absorb the light, send the electron to reaction center. Now from the reaction center, someone has to accept the electron. Now in the cyclic photophosphorylation, who is the first electron acceptor? Followed by and plastocyanin. You should not compare. Okay. Now here, in case of non-cyclic photophosphorylation, the first electron acceptor is what? Pheophytin. What is that? Pheophytin. This is most important. I want you to remember. Then followed by, can you see this? What is this? Plastoquinone A and plastoquinone B. Then cytochrome B6F. Same. Then plastocyanin. Same. I am just showing you to remember. That's all. In case of a cyclic photophosphorylation, the first electron acceptor is FD. In case of non-cyclic, pheophytin. Remaining all are same. Correct na? But in the cyclic, what happened? From the plastocyanin, electron came back to the origin. But since it is a non-cyclic photophosphorylation, it never comes to the origin. So can you observe? From the plastocyanin, electron reached to where? P700. What is P700? Photosystem 1. Very good. So again it absorbs the light, send the electron to where? Reaction center. Now reaction center get excited, release the high energy electron. So now who is going to accept it? Someone has to accept it definitely. Who are the electron acceptors? Remember, A0, number 1. Number 2 is A1. They are the name of the electron acceptor complexes. Followed by series of iron sulfur proteins are there. Iron sulfur X, iron sulfur A, iron sulfur B. Then FD, then FNR. You have to remember there is no shortcut over there. So from the P700, the first electron acceptor is A0. Next, series of iron sulfur protein FES, X, A, B. Then ferredoxin. FNR means ferredoxin NADP reductase. What is that? Ferredoxin NADP reductase. Don't get confused. So these are the various series of electron acceptors. Finally, FNR is involved in the production of what? NADPH. Correct na? Now very important question, all of you look at the picture and tell me, two photosystems works simultaneously or sequentially? What is the meaning of sequential and simultaneous? Sequential means one works first followed by the other. Simultaneously means both are working together. Correct na? Same. What you people can observe in the picture, P680 is absorbing the light. P700 is also absorbing the light. Correct na? Now there is no confusion. We will see that. Finally what we produced? NADPH. So what is our goal? To produce ATP and NADPH. Okay. Slowly understand, don't confuse with my words, understand correctly. This is what photophosphorylation cyclic happened in the bacteria. Do we fulfilled our goal of light reactions by using this cyclic photophosphorylation or not? Do we fulfilled the goal of light reactions by using cyclic photophosphorylation or not? First of all, what is our goal? ATP and what we produced here? Correct na? Because when electrons are moving, they lost some energy, then that energy can be used to produce, but what we have not produced. So only energy equivalent produced, reducing equivalent is not produced. Correct na? But here definitely we will synthesize. Where? In non-cyclic we produce ATP also and we produce, in this picture it is showing only production of NADPH. In the next picture we can produce ATP. So in general, in the non-cyclic photophosphorylation we produce both ATP also as well as the NADPH also. There is no need to get confused over there. Okay. So now what is my question I am asking you here? Focus. As we discussed that the two photosystems are working what? Simultaneously, which means P680 absorbs the light, at the same time P700 also absorbs the light. Now all of you tell me what happened here. P680 antenna pigments absorbed the light, send the electron to reaction center. Very, very good. Which means it is deficient of electron. Yes or no? Similarly, P700 antenna pigment absorb the light and send the electron to reaction center. Can I say P700 is deficient of electron? Who is going to replace the deficiency? Plastocyanin. Correct or not? Plastocyanin send the electron to the antenna pigment followed by the reaction center. So whatever the electron deficient in the P700 replaced by plastocyanin. Similarly, P680 send the electron to reaction center, it is also deficient of electron. So who is going to replace this? So this electron replacement for the P680 is mediated by water, which is called as what complex? The process called as photolysis of water. Correct now? What does it mean? Why we use the word photolysis of water? Very splitting of the water in the presence of light. That's why we call it as photolysis of water. Lysis of water in the presence of light. This photolysis of water is also called as what complex? Oxygen evolving complex. There is no confusion. Focus on the picture correctly. When one water molecule breaks down, we get 2 proton, 2 electron and half, not full. This is the oxygen what we breathe. And what happened to the electron? Just now we discussed, right? P680 is deficient of electron. So who will donate that? This water molecule breakdown. What happened to the protons? Protons released into the lumen. Where? Into the lumen. No confusions at all. Now once again observe the picture. Now who requires the electron? Who requires electron? P680. Because P680 is deficient of electron because it passed electron to the reaction center. Now if you observe the picture, of this particular P680, what does it made up of? This is the structure of P680. This is. Okay. If you observe the P680, it is made up of 4 proteins. CP43, CP47, D2, D1. There are 2 blue color and 2 green color protein. All together make up of P680. Correct na? This is the composition. No need to get confused there. Okay, now who is the deficient of electron? P68. Correct na? Now who is donating the electron? Water. Water molecule breakdown, we got electron, proton and oxygen. Now someone has to accept this electron, correct na? Who will accept this? So there are 4 proteins are there, CP43, 47, D1 and D2. This D1, what is that? D1. D1 is one of the protein of P68. This D1 is made up of an amino acid called as tyrosine. What is that? So the tyrosine amino acid present in the D1 protein of P680 absorb the electron from water and it transfer further. Correct na? So can I use this line? Tyrosine residue in the D1 protein of PS2 act as a final electron donor to the PS2. Correct na? I am only PS2, I need the electron. Who will accept the electron from water? Tyrosine will accept. Where the tyrosine is present? D1. So from the D1 electron passed to all other parts of the P680. You read in the 11th standard, if the water molecule wants to break, we definitely require manganese ions as the cofactor. Correct na? How many manganese? 4. And there is a requirement of calcium also, there is a requirement of chlorine also, apart from the manganese. But the major essential ion is manganese. Correct na? So by that water molecule will be breaking down. Now tell me, what happened to the protons? Electrons who accepted? P680. What happened to the protons? Protons are released into the lumen. Correct na? Now the question in the exam, where is the site of the photolysis of water? Lumen. And protons also released into the lumen only. Correct na? Don't confuse over there. Now the most important thing which we all has to focus here, what we discussed only, production of NADPH. What we have to remember, because in the non-cyclic photophosphorylation we can also produce ATP, but we understand only the production of NADPH. Now let's understand how does this ATP can be produced. Now all of you please answer by looking at this diagram. Which is the first electron donor, or simply understand, which is the first electron recipient from P700? Which is the first, look at the diagram, which is the first electron recipient from P700? Which one? A0. Correct na? Which is the final electron donor for the P700? Final electron donor is the plastocyan. Correct na? Think correctly and answer. Which is the last electron involved in the production of NADPH? Which, in simple words, which is the last electron acceptor involved in the production of NADPH? See, observe the picture. From the FD, who is accepting the electron? FNR. From the FNR again, electron is transporting to where? NADP+. Then we get NADPH. So the final electron acceptor is NADP+. It is not FNR. Okay, do not get confused over there. Okay, good, good. Everyone is correct? Okay, now we have to understand very clearly about the most important step, that is the process of ATP synthesis. Anyway, we produced NADPH. Which side? Stromal side. Correct na? Now focus all of you. Now tell me, no need to confuse. In the production of NADPH, which photosystem plays a major role? PS1 or PS2? Slowly, slowly. PS1. Because FNR is present here, right? What is FNR? Ferridoxide NADP reductase. Correct na? This is associated with PS1. That is only involved in the production of NADPH. Don't confuse. Similarly, which plays a key role in the production of ATP? PS2. Now let's prove how. Okay. Now this is the most crucial step. Very much tricky questions can appear from here. I'll explain it slowly. All of you focus on the picture correctly. Just now we discussed about the PS2. Correct na? I am explaining you how the ATP is getting synthesized. The process of ATP synthesis occur via a force called as PMF. What is called PMF? Proton motive force. Now let's understand how exactly it is happening. All of you focus. This is the P680 and it absorbs the electron, finally send it to reaction center get excited, release the electron. Who is the first electron acceptor? It is what phosphorylation? Non-cyclic. In the non-cyclic, pheophytin. Don't confuse. In the cyclic it is FD. In the non-cyclic, pheophytin. Very good. After the pheophytin, forgot. After the pheophytin, plastoquinone followed by cytochrome B6F followed by plastocell. So simple. Don't confuse. Now all of you pay attention, every one of you. From the PS2, electron is reached where, friends? First, pheophytin. Correct na? Now focus, all of you. Understand the concept correctly. From the pheophytin, electron reaches to where? Plastoquinone. Now every student pay attention, don't confuse. Upon accepting the electron from the pheophytin, plastoquinone reduced to PQH2. I hope you are getting my point. First, from the P680, who accepted electron? From pheophytin, electron transferred to? Now plastoquinone, upon accepting the electron from pheophytin, it got reduced to PQH2. But in order to become PQH2, it not only require electron, it also require proton. It accepts electron from where? Pheophytin. Proton from stroma. Stroma is rich of protons. So in simple words, PQ accepted electron from pheophytin. Proton from stroma. PQ reduced to PQH2. Wonderful. Then what happened? It never exists in the PQH2 form. What it has to do, tell me? It has to definitely transfer the electron. So it will transfer the electron to whom? Very good. It transfers the electron to cytochrome B6F. Then PQH2 oxidized back to PQ. You are getting the point. Then what about the proton? It releases into the lumen. Correct na? Once again understand the concept. Upon accepting the electron from pheophytin and accepting the proton from stroma, PQ reduced to H2. PQH2 again transfer the electron to and come back to PQ. Then it release the proton to lumen. Correct na? So all the protons moving from where to where? Stroma to lumen. And just now we discussed when a water molecule break down also we released proton. Now the concentration of the proton become higher in the lumen compared to the. Now what happened? All the protons, in order to maintain the charge balance, they started moving from lumen to stroma. Correct na? Whenever they are moving from lumen to the stroma, there is an important enzyme is located on the membrane, which is called ATP synthase enzyme. It is having two complexes, later we read in the biochemistry, F0 and F1. That F1 complex involved in the production of ATP. Now remember, proton always move in the form of a wave. Correct na? So remember, see, understand my statements correctly. When electron is moving, it lost energy. You're getting? When proton is moving, it generates energy. Now proton started moving from where to where? Lumen to stroma. Now finally it came from lumen to stroma via ATP synthase enzyme. Then what is generated? Energy. That energy is used by the ATP synthase enzyme to combine ADP with PI. Hence there is a production of. So can I say the production of ATP is mediated by proton motive force. Correct na? When the proton started moving, it generated energy. That energy can be used to join ADP with PI. Correct na? So proton motive force is involved in the production of ATP. So which photosystem is directly involved? PS2 or P680. In case of NADPH, PS1. Correct na? So you have to remember very clearly which photosystems are involved. Okay. Quickly answer, all of you. Online people answer. I don't know about the volume. I'll make it clear. Okay. Tomorrow also class happen through the YouTube only. I want every one of you to join the class without fail. Okay. So from tomorrow onwards, I'll go super fast. What is the answer? B. B. Okay. So they are asking about jet scheme. So jet scheme means it is what transfer? Non-cyclic photophosphorylation. In the non-cyclic photophosphorylation, first is P680, then followed by pheophyte. So definitely it is wrong. Correct, na? So P700 followed by A0, A1, iron sulfur X, A, B, FD, FNR, NADP plus. So this is right. Then P680, pheophytin, quinone A, B, cytochrome B6F, plastocyanin, P700 are also right. Correct, na? P700 after that A1, it is wrong, na? After that A0. So Q and R. Okay. So option will be the B. Okay. Answer this. D. See, because first is definitely, here they have not mentioned whether it's a cyclic or non-cyclic, but we know the electron flow. So from the P680, either it should be a ferredoxin or it should be a pheophyte. Correct, na? These two are differ. Then followed by common is what? Plastoquinone, cytochrome B6F and plastocyanin. So in none of the options they have not mentioned about ferredoxin or pheophytin. Then we have to consider the next electron acceptor only. Correct, na? This is so definitely wrong because this is the last one. Right. So P680, this is right PQ, but it cannot be PC. So P680, plastoquinone, cytochrome B6F and plastocyanin. Correct, na? This is the scheme of the electron flow. Okay. Okay, is it clear everyone? Okay. Till now the light reactions. Okay. Online people attend the class tomorrow without fail. Okay. Okay. See, from tomorrow the timings are same. Tomorrow is Tuesday, right? I am telling you the schedule up to Tuesday only. Up to Saturday. Okay, up to Saturday. So 9:30 to 10:30 will be the paper 1, or 10:45 or 10:50 I will start up to 1 p.m. Okay, I am telling you for this up to this Saturday. Okay, on this Saturday we have a PYQ series on whatever we complete. Okay, because from the next week, as I said, I will take the evening class also up to 2 to 4 p.m. Okay, as a result, in the span of 1 month only we will cover 4 units. Okay, it's everything depends on you people. Okay, done. Okay, all right, everyone attend sharp 9:30 a.m. tomorrow. Tomorrow also it happens in the YouTube only. From day after tomorrow it will be in the app. Okay, all right, all right. Okay, come tomorrow sharp 9:30. Okay, inform others who are in the batch. Okay, tomorrow class has to become filled.